Hi, I'm Abhi.
I'm Chloe.
And I'm Alex.
Oh, this is photosynthesis. The equation for photosynthesis is 6CO2 plus 6H2O, which becomes C6H12O6 plus 6O2.
This is our magnified version of a chloroplast, which is an organelle inside of a leaf. So here, sunlight and water go into the thylakoid and the thylakoid space to produce oxygen. On the other side of the chloroplast, CO2 or carbon dioxide goes into the Calvin cycle and produces glucose. NADPH cycles through and becomes NADP plus and goes back around again to become NADPH again. Same goes for ATP, which cycles through and becomes ADP.
Then in the stroma is where the Calvin cycle takes place. This process is when small pores on the surface of the leaf take in atmospheric CO2, which is then paired with the carbon from 6 5-carbon molecules to create 12 3-carbon molecules, 2 of which are used for processes like sugar synthesis. The rest of the 10 3-carbon molecules are then recycled to become 6 5-carbon molecules again.
Hello, I'm Kevin.
I'm Aditi.
I'm Abhi.
And I'm Angela. And this is cellular respiration. How does energy flow? Energy enters plants through sunlight and is then used to create glucose. Glucose is then consumed by heterotrophs, which gain their energy from autotrophs. The overall equation of cellular respiration is 6O2 plus C6H12O6, which becomes 6CO2 plus 6H2O and ATP. Glycolysis is the first process of cellular respiration. It is an anaerobic process located in the cytoplasm. It starts with glucose being broken down into 2 pyruvate molecules. 2 ATP are invested into this process and 4 ATP are produced, giving a net total of 2 ATP. Lastly, 2 NAD plus is given a pair of high energy electrons and a proton to become 2 NADH, which is safe for the ETC.
The second process of cellular respiration is called the Krebs cycle. The Krebs cycle, also known as the citric acid cycle, takes place in the mitochondrial matrix. The Krebs cycle uses pyruvates from the glycolysis and pairs them with other molecules to create citric acid and a carbon dioxide. The citric acid is broken down and cycled through the Krebs cycle to create high energy electrons, which are paired with NAD plus and FAD to create FADH2 and NADH. These high energy electron carriers are sent to the ETC to create ATP.
The last process in cellular respiration is the electron transport chain. High energy electron carriers drop off their electrons, which are then used to pump H plus ions from the matrix into the intermembrane space. The concentration difference then powers the movement of ATP synthase, which binds ADP with the phosphate group to create ATP. For every glucose molecule, around 34 ATP are created.
In the absence of oxygen, organisms produce ATP via fermentation. The purpose of fermentation is to replenish NAD plus to continue glycolysis without oxygen. There are 2 types of fermentation, alcoholic fermentation and lactic acid fermentation. During alcoholic fermentation, pyruvic acid is broken down into 2 CO2, 2 ethyl alcohol, and 2 NAD+. During lactic acid fermentation, pyruvic acid is broken down into 2 lactic acid and 2 NAD+. The NAD+ cycles back into glycolysis.